Every year, cancer claims around 10 million lives worldwide. We have different powerful treatments. We have chemotherapy, we have immunotherapy, and we also have targeted therapy. However, in most cases, these therapies tend to eventually stop working. The reason why this happens is because of drug resistance. This means that the cancer cells eventually start to evolve and they also start to adapt. This brings us to 90% of failures or treatment failures in metastatic cancer happen because of drug resistance. A tumor is not just a lump of identical cells. It's basically an ecosystem. What this means is that we have different populations of cells which live together. They compete for space and also nutrients. The thing is that in these populations, there are some cells which actually are affected by the treatment and also the drugs. We call these the sensitive cells. However, in these populations, there's also a type of cells that are not affected by the drugs because of their type of mutation. The critical thing here is that they just don't depend only on the drugs, but they also depend on each other. When we apply aggressive chemotherapy, we kill most of the sensitive cells, but in doing so, we remove the competition. We clear the way for resistant cells to take over. Researchers have actually developed a strategy called adaptive therapy that tries to work with this competition. Instead of maximum dose, it uses careful strategic dosing to keep sensitive and also resistant cells competing, using evolution against the tumor. But to do this effectively, we need to know how these populations behave. We need to understand the rules.
So how do we find these rules? Right now we have two main approaches, and both of these approaches has its own limitations. First, it's traditional mathematical modeling. Scientists write down equations based on biological theory. But this requires us to assume we already understand what's happening, and cancer is complex. Our assumptions may be wrong or incomplete. The second approach is machine learning. Neural networks can learn patterns directly from patient data without any prior assumptions, but they are black boxes. They might predict accurately, but they can't explain why. And we cannot tell a doctor to rely on these based on the algorithm said so. So what we need is something that learns from data like machine learning, but also gives us transparent, interpretable rules like additional models.
And this is exactly the problem that we're trying to tackle. We're trying to build an interpretable pipeline where we're trying to learn how these cancer sets interact and their dynamics in a way that's interpretable, and at the end produce equations that best represent it. At the core of this approach is a special type of neural network called Kolmogorov-Arnold Network. What makes it special is, unlike traditional neural network where we have fixed activation functions and at the end we produce a lot of weight vectors that's not interpretable, what we have is a learnable activation functions that are quite interpretable, and we can zoom in, open up, and see how this model is reading or understanding the data, and which is quite important, especially for clinical applications where life is at stake. Once we train the model, the next step is what we call a symbolic regression, a step where we best approximate what was learned from the model into a human readable equation. And this equation best represents the dynamics. And we're not just getting one equation, but candidates of equations. But not all of these candidates make sense. A model might fit the data perfectly, but what was learned may not make sense biologically or in a game theoretic way. Which is why we test each candidate using biological and game theory criteria. Some of those criteria include: does it prevent negative growth? Under drug presence, does it capture the expected cancer cell response? And does it capture realistic cell interaction? And does it respect carrying capacity, which is the natural limit on how large a population can grow?
Currently, we are validating our pipeline on a test system, which is the Lotka-Volterra equation. And this equation describes how two types of population grows, compete, and interact with each other, just like resistant and sensitive cell acts in cancer. Early results are encouraging. The pipeline recovered the correct mathematical structure, including the right equation forms and population interactions. Right now, we are focusing to improve our accuracy, and then we will move to the real experimental data, which is in vitro non-small cell lung cancer data, which contains the population measurement of resistant and sensitive cells on different experimental setup.
We're not just trying to predict where the cell population is going to be in time, but we're trying to understand how and why it does what it does. we're trying to understand and uncover the rules that govern the dynamic.